Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
All right, can you see my screen here? So, welcome. This is the test driving valid session, number 2 in this case. If you have been at the September 2nd meeting, you know, we have sort of put together or put this online as sort of the results and or notes of the initial session. And, you know, sort of what would be sort of the use cases. And so I don't know if you had time to take a look at this, but I think we can probably work or continue our work on these 3 use cases. I think for today we had the Pdl1 use case identified as sort of like a more AP-based solid tumor companion diagnostic identification. And in this particular instance, that would be cross-referencing another approved test. So I thought that would be, you know, a good starting point to discuss this. Yeah. So how do you think we should approach the topic?
So just a definition of cross-reference from the legislation, because it's quite broad in my opinion. It's referencing in its labeling, so that could be in your test catalog, for example, the name or intended use of another medical product that is not an in vitro clinical test. So referencing another test wouldn't matter, but referencing another product, so for example, a drug. So if you said, you know, a positive Pdl1 means that this patient should be able to take XYZ drug, that would be potentially considered cross-referenced.
Carla. Hey.
Hello.
Hey, Rashni. Sorry, I didn't see you at first. Carla, congrats.
Thank you.
All right, we already started. Today's topic is the Pdl1 immunohistochemistry, and Shannon was just sort of explaining what cross-referencing in this context sort of refers to.
So it's essentially getting at the companion diagnostics question.
Mm-hmm. So this would effectively mean there is sort of like the starting point for the companion diagnostics would be there is a drug test couple that is authorized, right? And we make a claim that our, meaning the to be validated or to be certified Pdl1 test can cross-reference this prior authorization, correct?
Well, essentially, you'd be stating in your intended use, our PDL1 test will determine whether your patient would be successful taking a particular drug and naming that drug. I think that would meet that definition of cross-reference. We're cross-referencing to another medical product.
Like this?
Yes.
So this would be the intended use, right?
Yes.
So I think let's dive right into our sort of rubric here, right? So is it low risk, is it high risk, or is it high risk with mitigation measures?
Well, this one is tricky too because this is a stain, right? So what is the test? If you've got a clinical chemistry assay that you're running on a mass spec, the test is the mass spec, right? For a lot of these stains, the test is the pathologist's eyeballs. So that's where this gets a little bit complicated too. And at what point do we kind of segue into practice of medicine, which VALID is not intended to regulate?
But the procedure, right? So VALID would apply to the procedure, meaning not just the IVD, here the Pdl1 antibody and the stain, but how the pre-analytical variables and the interpretation sway the results, right?
Yes.
So I think this gets into the tricky issue of, let's say, this is the, I don't know, if this is the IVD and this is the lab, sorry, not easy to do this here like this, but so question would be, how would a lab that claims that we're able to identify the people draw a line between the functioning of the PDL1 and the procedure to, execute the report? Because I think that was, I think the word procedure is included in the definition, right, of the IVD, or of the service, I think is what they use. Let me just double check.
So you're right. So preparation is included in the definition of an IVCT.
So I think that is the biggest part. So, the lab will obviously have other components to it, but I think that would be a fundamental change that, we have to draw a line somewhere, like which components of the procedure would account or would have to be accounted for in the submission and in the, establishing the performance of this test.
And there's also the other wrinkle that, again, we're talking about an AI application here, but if this was a manual test, then, that's exempt under VALID. And so, that's, I don't know, I often think of IHC as kind of a manual process, right? Having a person review that and interpret it.
And why would this be exempt?
There's just an exemption for manual tests.
Got it.
Says
if you throw it on an
vitro.
Auto. Sorry, go ahead.
Oh no, I was just going to say. I'm just reading. Is the result of direct manual observation with or without the use of automation I'm sorry, without the use of automated instrumentation or software for immediate or final interpretation by a qualified laboratory professional. So if you're not using an automated device to produce the final interpretation, then it's exempt. And my interpretation of the way VALID was written is that is specifically for IHC and a few other direct observations in clinical medicine and laboratory.
Mm-hmm. So then, of course, sorry, Shannon, but what did you say earlier about the risk? It would be probably higher risk, right?
So go ahead.
I think
Oh,
I
sorry.
think it depends on how much we expect the pathologist to interpret, right? If the pathologist, depending on what the AI is doing here, right? If the AI is kind of coming up with the answer, the pathologist is not interpreting a lot, so that would be higher risk because there's no, correction. Whereas if it's just informing the pathologist that the pathologist makes the ultimate decision, it's probably like moderate risk. Yeah, it just depends on what the product does, right?
So let's say if AI, we would probably all agree. So if AI makes the decision, it's probably highest risk, right?
Yeah, that's like a class 3 device.
And then if it's decision support, and let's say the algorithm would provide a score of percentage positivity, but a human would sign it out, that would likely be with mitigating measures, right?
Yeah.
So I don't know, do we have an abbreviation for this? No. The third tier. Yes, I knew there was something there. Decision support would be the third tier. And then manual, like let's say a pathologist has to interpret it, we would say it wouldn't apply, right?
The risk there is that they get it wrong, right? But that's...
Yeah, but it's exempt from VALID, right? So if it's a manual stain and a manual interpretation by a human, it would be exempt from VALID, correct? At least, I don't know, but...
I think that's fair. In a previous version, I think what was throwing me